Hi there, part 2 of the second unknown project. Right, it occurred to me that there are still a couple of things in the lab manual that I didn't cover. So I'm on page 229 now, and on the top of the page, number 4, it says that if you should contaminate your culture or for any other reason require a replacement of your unknown, the cost is 30 points. Here's what I mean by that. If you should contaminate or break your working culture, and some other horrible thing befall your stock culture, then pretty much game over. It would cost 30 points for a replacement, and you only had 35 points left, so that's not going to be a really good situation. But I have to tell you that in all the years that I've done this, I've really never had that happen. Okay, so this is a point where you go to the library and you start to gather information from the Bergey's manual. Due date will be on your syllabus, and we'll discuss that as well. And please keep in mind that you will not be able to begin any of the biochemical tests on your flow chart until that flow chart is approved by me. Okay, now when you write your flow chart, and this is in the lab manual as well, I want you to avoid using these tests, and that would be motility, capsules, and pigment. So those, I call them jokingly the forbidden 3 tests. Here's why. Even if Bergey's manual reports, let's say for motility, that the organism is positive, and you could replace motility with capsules or pigment, those results are highly variable, and it would be too easy to try to make a decision based on the results of one of those tests and be wrong. So avoid using those tests. There is only one acceptable exception to that, and that would be when you're writing your flow chart, when you get to the point where you have, let's say, just 2 organisms left to separate, and there is no other test available except one of those 3, then it's okay to use one of those tests. If you use them anywhere else in your flow chart, anywhere higher up in the flow chart, I will deduct a significant number of points. Okay, now let's talk about what tests are available for you to use and how to dig information out of the Bergey's manual. Okay, what tests are available for you to use? That one's pretty easy. Anything that's in the lab manual, that's what's available to use. Nothing else. Those are the only tests that we have available in this laboratory, and I have really tried to design a project that works with those tests that are available. Now, if you're a 102 student watching this, there are several tests that we do in 150 that we don't do in 102. Those tests are available for you to use as well, and if you just remind me that you haven't run that test, we'll either myself or Mrs. Burns or Renee will help you, we'll kind of give you an overview of how to run the test and how to interpret the results. Okay, so you've got this checkerboard matrix. You're going to be taking that down to the library. You've already written all of the organisms' names that were on your bug list here, and then you've got your the names of the tests up above. So let's talk about how you get the information that you need out of the Bergey's manual. Well, the first thing you need to do, and you should have a photocopy of a page of Bergey's manual in front of you. The first thing you want to do is determine the gram designations of all of the organisms on your unknown list. That would be a good thing, and you can record that on your checkerboard matrix. And then look them up one at a time by their genus name in the table of content. I have found the table of content to be more user-friendly in Bergey's manual than the index. The reason I say that is the index is so exhaustively referenced, I mean down to tiny little minute details, that if you try to follow all of the references in that index, it's going to take you on a wild goose chase. So look the organisms up by genus name in the table of content. So the page that you have in front of you, one of the organisms on it is named Klebsiella Pneumoniae, Subspecies Pneumoniae. So let's go ahead and look up some information about that organism, and let me identify for you the main places in the manual where you're going to find what you need. All right, so first thing is I determined that Klebsiella is gram negative. And I recall from my bacterial survey that it's a member of the group known as the facultatively anaerobic gram-negative rods. Okay, great. Then I go down the list and I find Klebsiella. All right, so I'm going to turn to that page. Happens to be page 181 in Berge's manual. And I'm going to take a look at, if I can find the page, I'm going to take a look at, there is a big, fat, juicy paragraph here. I'm sure you can't see this, but I'm going to do this anyways, that says Klebsiella, and it gives a description of the genus. Now, I'm primarily interested in the information that's in boldface. There is way more info in Berge's manual than I think any human actually needs, but I'm looking mostly at the bold-faced information, and it says, let's see, facultatively anaerobic, form capsules, don't use that one. Oxidase negative, I can use that. Catalase positive, I can use that. Arginine, decarboxylation of arginine negative. All right, so there's several little bits of information there that are useful to me. Then what I'm going to do is I'm going to flip through the section and I'm looking for the tables. And you've got a copy of one of these pages. This is the meat and potato of the information that you're going to require for writing your flowchart. So anyways, you will see the organisms' names listed along the top, and let me find that page that has Klebsiella on it. I think I need the old glasses here. And here it is, Klebsiella pneumoniae, subspecies Pneumoniae. The organisms' names are along the top of the table, and then the test names are along the axis on the left. And let me just kind of run through this, and you can be looking at your handout while I'm doing this. All right, so it says Klebsiella pneumoniae, subspecies Pneumoniae. It's gram-negative, it's oxidase negative. Methyl red is variable, okay, because what I'm seeing reported for the methyl red results is a negative sign in brackets. Okay, I'll come back and I'll talk about what variables mean in just a couple of minutes, or I should say how variables are recorded in Berge's manual. It was indole negative, let's see, VP positive, citrate positive. Terrific, so I'm finding lots of information about Klebsiella pneumoniae, subspecies Pneumoniae here in this table, and I'm recording that information on the checkerboard matrix. Anything that you find in Berge's manual as a variable, right, let's talk about that for a minute. Now, if Berge's manual reports a test as just plain old positive, I mean no brackets around it, no disclaimers, then it really is positive. Or if it just says plain old negative, then it really is negative. But if Berge's records the results as, let's see, positive in brackets, negative in brackets, the letter V, the letter D, either capitalized or lowercase, or occasionally you'll see ND for no data. Treat all of those as variables, all right? And I would say just record them as a V in your checkerboard matrix. Okay, so the only tests, like I said, that are available are the ones that are in the lab manual. And let me kind of run through this list of tests and just make sure you understand what each one of them is. Okay, back to the top again, gram stain, that's easy. Oxidase test, I think that one's pretty easy. Indole, we did that. Methyl red, that's the MR test. Vogts-Proskauer, that's the VP test. Hydrogen sulfide production, that's the test we did using triple sugar iron auger. Urea hydrolysis, we demoed that one for you. That's the one where we use the rapid urea auger, and it changes from kind of a peach color to a fuchsia color for a positive. Phenylalanine deaminase, that's the phenylalanine test. Okay, now the vocabulary here in Berge's is slightly different than what I used in the lab manual, but lysine, arginine, and ornithine, those are the decarboxylation tests that we did. Motility, we're going to avoid that one. Gelatin hydrolysis, we do not have that test. KCN, we don't have. Malonate, there's a test that we have. Now, take a look at the next several entries are fermentation of carbohydrates, beginning with D-glucose, and that's the glucose that we used. Now, the only one that's broken into an acid line and a gas line is glucose. So acid means, did the broth change from red to yellow? That's the reaction we're interested in. That's the line that you want to use in Berge's manual if you do need to use the glucose fermentation test. The gas line, that would mean was any carbon dioxide gas captured in the Durham tube. Highly variable. Don't use it. It isn't going to help you. All right, so D-glucose, that's the glucose we used. Going down that list, lactose is there. Maltose, we do not have maltose, and that has nothing to do with the starch test, so just ignore the maltose. D-mannitol, that's the mannitol test we used. And then finally for carbohydrate tests, the sucrose test. Right, continuing down the list, the nitrate reduction test. That's the nitrate test that we did in lab, and that's just the first part of the test. Was the organism able to convert nitrate into nitrite? All right, so that's what we mean by the nitrate test. Deoxyribonuclease at 25 degrees Celsius, that's the DNase test. Not typically very useful for gram-negative unknowns, might be useful for gram-positive unknown. Let's see, and then catalase production is down near the bottom of the page, and then oxidation fermentation test. If you have a gram-negative unknown, I don't know that that one's going to be terribly useful to you. Pretty much all of them are going to be fermentative for that test. Okay, so anyways, you're going to get information from the Berge's manual, transcribe it onto your checkerboard matrix, and then you're going to be ready to start writing your flowchart. So let's talk a little bit about that now. Okay, we've done a little bit of work writing flowcharts. We did some in class together, and you've got a couple of practice exercises that I'm going to ask you guys to do just to make sure that you're clear on how to do this. But you have to make a decision to get started, and that is, what test do I want to begin my flowchart with? Now remember, you guys, I told you that there's always more than one correct way to write a flowchart. So I would try to begin my flowchart with a test that doesn't have any variable results in it. If you can do that, that's great. You may not always have that luxury, but if you can, that would be the way to start. Okay, this video is getting a little long, so I'm going to go ahead and break and make a third segment to wrap this up. Thanks very much.